This method provides researchers with a new way to prepare aquatic plant roots, the surrounding rhizosphere, and bulk soil for elemental imaging techniques. The main advantage of this method is that it's relatively quick and affordable, while also accurately preserving the distribution and speciation of elements of interest.
This technique can be used to answer questions regarding many elements in plant systems, from nutrients in agricultural systems to fate and transport of contaminants in phytoremediation systems. Be sure to work quickly while also being mindful of the desired area or areas of the root system for sampling, and take multiple samples per root system.
To prepare the SLAM freezing equipment for an experiment, place two 5 by 5 by 15 centimeter copper blocks horizontally into a clean cooler capable of holding liquid nitrogen. And, wearing the appropriate PPE, pour enough liquid nitrogen into the cooler to submerge the blocks. Once the bubbling subsides, place one spacer on each end of one copper block. Then, using tongs and cryogenic gloves, stand the other copper block on its end to make retrieval easier when the sample is in place. To collect the sample, either use a potted plant or use a shovel to begin extracting the desired plant and rhizosphere from the wet soil, taking care that the dug hole is much larger than the desired root volume. Use a steel blade to cut away any excess soil, taking care not to disturb the soil within the desired area. When the desired area is reached, cut an approximately 3 by 3 by 2 centimeter root cube, and immediately place the cube.